All right, welcome everybody to our November 1st installation of the UCSB Fall 2022 Virtual Seminar Series. And oh boy, just like our other weeks, we have a great talk for tonight. And in the spirit of Halloween, I might even call it a creature feature, but I'll let you all decide that at the end of the talk tonight. Before we get into it, I wanted to start out with just a little bit of housekeeping, just explain how the night will go. I'll talk a little bit about the Natural Reserve System, and then I'll kick it over to our speakers for tonight. So thank you again for joining us. My name is Marianne Whitman. I'm the Executive Director of the UC Santa Barbara Natural Reserve System, and I get the pleasure of being your MC for the evening. For tonight, the format for this evening will go such that Carol Blanchette, who's the director of the Valentine Eastern Sierra Reserves, will first tell us a little bit about the reserves and what's going on there. And then she'll introduce our speaker, Professor Tobin Hammer of UC Irvine. And Toby will give us a great talk tonight for about 20-25 minutes or so. And then after Toby presents, then we're going to open it up to a Q&A session. So this is where you all get to interact with us. And the way that you do that is you go ahead and look at the bottom of your Zoom screen, and you're all probably very much expert at this by now. And there's a Q&A button, and that's where you can click on that and submit your questions. And then we will respond to your questions in real time tonight. So very much looking forward to your feedback and how this evening will go. This tonight's talks will have closed captioning enabled. So if you would like to enable that on your end, you can use the button at the bottom of your screen again to enable live transcript. Tonight's recording will also be included on the UC Santa Barbara Natural Reserve System YouTube page. So if you want to watch it again or share it with your friends, then that will be available to you. That link will be sent out to you after tonight's talk. All right, so first, I wanted to earlier this quarter, I gave a little shout out to the UCSB undergraduates out there who are just starting their quarter. Tonight, I just wanted to give a shout out to all the graduate students out there. You're probably, midstream in the middle of a busy quarter or busy semester, taking classes, doing your research, trying to figure it all out. And it's a lot. And we all appreciate what you do. And we appreciate you for joining us tonight. Wanted to tell you also a little bit about the Natural Reserve System. So tonight, we're coming to you from the UC Santa Barbara Natural Reserve System. And the Natural Reserve System is a system-wide program in the University of California system. There are 41 sites distributed across the state, and they're set aside especially for research, university-level education, public service, and land stewardship. And what we get to do is the best thing in the world. We really get to enable and facilitate education and learning and research at these sites. We really try to learn about them and share that information with you all. So tonight, some of our presentation will be just that. So we really hope you will enjoy. UCSB oversees 7 of the 41 reserves, and tonight we're coming to you from the Valentine Reserve up in Mammoth. So with that, I'm going to go ahead and turn it over to you, Carol, and why don't you take it away from here?
All right, great, Marianne, thanks. And I will share my screen. And Marianne, just give me a thumbs up if that's good. You can see that. Okay, excellent. Hi, everyone. Thanks for tuning in. I'm really happy to introduce you to one of my favorite places, which is Valentine Camp. And for a large part of the year, the reserve here is blanketed in snow. And we're super excited tonight because here in Mammoth, it is snowing, it's dumping. So hopefully we're going to wake up to this scene tomorrow. And I'll give a shout out to the California Ecology and Conservation class tuning in from over at the Page Center tonight. Saw them all running around out in the snow, so they're pretty excited it's snowing too. Okay, let's see. There we go. Okay, so Valentine Camp is located on the eastern slope of the Sierra Nevada mountains in the town of Mammoth Lakes. And it's comprised of 155 acres. It's been protected from entry and grazing since the early 1900s. And it features remarkably pristine subalpine habitat, including montane forest, montane chaparral, Great Basin sagebrush, high montane riparian vegetation, wet montane meadow, and seep and spring vegetation. For those of you who are familiar with the town of Mammoth Lakes or Mammoth Mountain, Valentine Camp, if you can see this, lies here along the southeastern edge of Mammoth Mountain in between the town of Mammoth Lakes and the Lakes Basin. And the Valentine Camp property has a really special and unique history. It was purchased around 1916, just after the turn of the century, by a group of businessmen from Los Angeles. And they used this property as a hunting and fishing camp for many years. All of the cabins at Valentine Camp were built around 1920 and were constructed using traditional techniques called chinking, so real log cabins. And these are all still in use today by students and researchers. So here's one of those cabins. This is the university cabin. Visiting Valentine Camp often feels like you're taking a step back in time. So all the cabins, like I said, are over 100 years old, and the land preserves a remnant of what the area looked like before the town was developed. Over the years, several of those original owners, the businessmen, either sold out or transferred their ownership to William Valentine, one of the original owners, and his son, Ed. And eventually the Valentine Camp was left to Mrs. Carol Valentine, who was Ed's wife, and the Valentine Foundation. Mrs. Valentine, pictured here, was a great patron of the arts, and she had a real love for botany and horticulture. She was instrumental in the development of Lotus Land, for those of you in Santa Barbara, may be familiar with that zone. And she really understood the botanical significance of Valentine Camp. So at the time she had this property, she approached Vernon Cheadle, who is then the chancellor at UC Santa Barbara, also a botanist, with the idea of donating Valentine Camp to become part of the newly formed UC system of natural reserves. And it became officially a reserve of the UC NRS in 1972. So just for those of you out there who are doing the math, this year we are celebrating our 50th anniversary of the Valentine Reserve. So, and according to Mrs. Valentine, donating Valentine Camp to the UC NRS was the best possible way that she felt to preserve the land and use it beneficially. So the cabins at Valentine Camp are a well-loved haven for many of our researchers that come to study and conduct their research projects in and around Valentine and the Mammoth region. It's been used by researchers studying topics ranging from plant biology and forest ecology to wildlife. And we also have a fantastic group of citizen scientists who have been collecting and amassing a long-term data set on the phenology, or the timing of flowering and leaf production, for a variety of plant species at Valentine. And as you might hear about in Toby's presentation, understanding when certain species of plants are flowering can link to patterns of activity in pollinator species like bees. So we also host a wide variety of university classes at Valentine in fields ranging from biology to geology. The location of Valentine Camp, just below Mammoth Pass, also ensures that it gets a huge amount of snow each winter. And given the rustic 100-year-old nature of these uninsulated cabins that are fully buried under snow each winter, we only operate Valentine Camp for use by researchers and classes in the summer months. Approximately a third of Valentine's 155 acres is composed of a mixed conifer forest. And similar to many forest ecosystems of the High Sierra, fires have been suppressed in this system for the last 150 years or more. And this has led to an unnaturally dense forest that's no longer resilient in the face of disease, pests, and wildfire. And so in the last few years, we've undertaken a large fuel reduction project on about 50 acres of the high density forest at Valentine. And the goal of the project is to increase the health and resilience of the forest and decrease the risk of fire, not only to this site, but to the town of Mammoth Lakes. It's also providing unique research opportunities for us to improve our understanding of how these management strategies can affect forest health and wildfire risk. The 155 acres at Valentine are home also to a wide variety of fauna. Mule deer, shown here, are a common sight. And Valentine is really well known for its bears. Bears utilize the site throughout the season and den around the site in the winter. And although it's rare to see large cats like this during the day, mountain lions are commonly seen on our trail cams out there at night. We have a really active outreach program at Valentine, and we offer public tours for adults throughout the summer. And we're most well known for our outdoor science education programs for children. So elementary school kids from grades K to 5 come to Valentine every year for grade-specific field trip lessons and activities. And we host over 2,000 school children each year from across Mono and Inyo counties in our programs. And there's a huge body of literature demonstrating the importance of this sort of outdoor learning for children. And so Valentine really provides an ideal classroom for these programs and takes advantage of the wide variety of habitats to let children explore and learn about the environment. We also offer week-long camps at Valentine in the summer. And these are focused on topics ranging from natural history to photography to the arts. We also have an education center facility at Valentine, which was constructed in 2000, thanks to the generosity of many of our donors, and also provides an indoor space for learning and creative activities. All of this and all of our outdoor science education programs are also heavily supported by donations, and in particular, a really generous group of supporters called the Valentine Reserve Fund. And we could not do what we do without this support. And please reach out to me anytime if you're interested to learn more about that. So before I introduce our speaker this evening, I'm guessing that some of you in our audience out there are bee enthusiasts. And I want to take this opportunity to let you know about a really great book that's just been published. And some of you may have heard the interview with the author on National Public Radio a few months ago. So bees are amazing, of course, and a good deal of what I know about bees so far, I've learned from this book. So if any of you are looking for a nice holiday gift, I would like to recommend this book that's just come out and has been illustrated by our very own snarl scientist, pictured here, Alexa Lindhauer. Alexa studies frogs, but she's amazingly talented in all aspects of science and art. And it's really a richly illustrated children's book that tells the story of a bee that doesn't look like what we all think a bee should look like. And in fact, there are over 20,000 species of bees, and the story highlights and celebrates diversity in all its forms. So it's called Am I Even a Bee? And you can find out more at their website, amivenabee.com. And you can purchase this book online at Amazon, or even better, through your local independent bookstore. So that was our commercial. And I would like to move on to the main event here and introduce our speaker tonight, Dr. Tobin Hammer. And one of the highlights of my job is really learning about all the sorts of unique and interesting aspects of research that happen at sites like Valentine and Snarl. And before meeting Toby, I, like probably many of you, had never really thought about the gut microbiome of bumblebees. But as I found out more, the more you learn, the more interesting it becomes. And so Toby is just a great, great speaker, and he was so patient at explaining all of his research to me here one day at Snarl. So I'm really excited to hear about his work. He's an assistant professor of ecology and evolutionary biology at UC Irvine. He got his Bachelor of Science at UC San Diego and his PhD at University of Colorado Boulder. And the title of his talk tonight is Charting the Microbial World Within California's Bumblebees. Okay, so Toby, I will leave it off to you.
All right, thank you, Carol, for the introduction. All right, hopefully you can all see this. So thanks to all of you for being here tonight. I'm really excited to tell you about microbes and bees and some of the research that my lab at UC Irvine is doing to study the bee microbiome. So I wanted to start off by talking about microbes. What are microbes? They are basically just organisms that are too small to see with the naked eye. You need a microscope to see them. And there's actually a very broad range of different kinds of microbes. They fall into these three general groups. We have the bacteria, shown here by Yersinia pestis, which is the bacterium that causes bubonic plague. There's also a lesser known group of microbes called the archaea. They tend to live in extreme environments, but they're also in our own gut, like Methanobrevibacter smithii. And then there's also a whole range of single-celled microscopic eukaryotes. So like us, they have cells with nuclei. They're very distantly related from bacteria and archaea. They're also very abundant, very numerous, very diverse. And probably the best known species in this category is the brewer's or baker's yeast Saccharomyces cerevisiae. So I'm only showing 3 species here, but actually there are millions of species of microbes, and the majority of the evolutionary history of life on Earth is microbial. So the last common ancestor of all life existed somewhere around 4 billion years ago, and it remained microbial for billions of years. And only recently have the organisms that we're most familiar with, animals and plants, evolved. And we really make up just like a small sliver of the whole tree of life, shown here by those red twigs. And microbes are everywhere. They are super abundant. If you had microscope glasses, if that existed, this is more or less what the landscape would look like. There's bacteria and fungi and archaea in the soil and the plants and on the bodies of animals, inside the bodies of animals. It's been estimated there are around 1 nonillion microbial cells on Earth, which is a 10 followed by 30 zeros. Kind of hard to imagine, but you know, there are lots of microbes. And one of the most interesting places that scientists are studying the microbial world is in the human body. It's been estimated that there are about the same number of microbial cells as human cells, but we have a very diverse microbiome. We have hundreds of species of microbes, and they vary a lot in what kind of genes they have. So if you look at the kind of repertoire of genes in our bodies, the vast majority of that is actually coming from microbes as opposed to our own cells. And collectively, all of these microbes weigh about 200 grams, or about the same weight as your pancreas. And I kind of like to think of them as an organ, kind of like a hidden organ that we're only recently starting to understand. And it has lots of important jobs in our body. It's really crucial for sustaining our health. And it provides us, because we have so much diversity and all this kind of genes that we can acquire, it provides us with a lot of flexibility. And I would say that we are currently, the field of biology is kind of in a golden age of microbial exploration. So if you think about like the Renaissance era to the Victorian era, where people were going out in ships and charting distant lands and using new telescope technology to study the heavens and collecting birds and plant specimens, we're kind of in that mode right now, but for microbes. And we have different technology. Instead of telescopes and you know, collecting nets and so forth, we have DNA sequencing. And this is really important because, as I showed you previously, a lot of microbes look pretty similar. And you might have two kind of yellow rods, like are shown here, that could be as distantly related to one another as you are from a moss or a mushroom or something. So it's really hard to distinguish species based on what they look like. So this is where we turn to DNA sequencing. And the sequencing technology, which is the same that's been used for sequencing the human genome, has really revolutionized this field of microbiome science, really opened up this world of microbes to exploration. In my lab at UC Irvine, we primarily study the diversity and functions of the microbial world associated with bees. So I wanted to give you a little bit of a primer on what bees are, some facts about bees. The first one is that bees are basically vegetarian wasps. So they evolved from an ancestor that was predatory, fed on insect prey, and they figured out how to make a living on pollen and nectar. Almost all bee species completely rely on flowers as their sole source of food. They get their sugar, their carbohydrates from nectar, their protein and lipids from pollen. They're very diverse. As Carol mentioned, there's about 20,000 species or so globally. California has about 1,600. This is a very good place for bee diversity. So if you live in California, Congratulations, we have a great bee fauna here. And most of them are solitary. So if you're most familiar with honeybee, lives in hives, tens of thousands of individuals in these dense hives. That is not what the lifestyle of most bees is like. Most of them, there's a single female. She's digging a nest or creating a nest in some kind of substrate. She's provisioning with food, laying eggs. This is the lifestyle of most of these bee species. And of course, they're crucial pollinators. So around 70% of the crops that we eat are bee pollinated. If we lose those bees, there's going to be a lot of problems for agriculture. And we do seem to be losing bees. So you may have heard about bee declines. There's a lot of stressors that bees are facing. This includes climate change, diseases that we are helping spread, pesticides that we're putting onto the environment, and also lack of flowers, which again is their sole source of food, and a lack of places to nest. And a lot of these threats synergize or interact. So for example, if you're a bee that's being poisoned by pesticides, you're less able to fend off disease. So there's a lot left to be learned about which of these stressors are most important and how they interact, but this seems to be kind of the main ones that are harming bee populations. And then of course, bees are home to microbes, which is what we study. Our primary focal group of bees right now are the bumblebees, the genus Bombus. These are particularly vulnerable to stress for a couple reasons. One is that they're kind of large, they're furry, that makes them not very well equipped to deal with heat. And we know that climate change, heat stress is strongly affecting bumblebee populations. They're also social, kind of like honeybees, but with smaller colonies. But because they're social, they can't do social distancing. It's very easy for, if a single bee comes back to the nest with carrying some kind of infection, it's very easy for that to then spread. And we have 47 species of bombus, of bumblebees in North America, north of Mexico, and around a quarter of them are either vulnerable, endangered, or critically endangered. So there's a lot of issues right now. A lot of researchers are trying to figure out what is causing declines and what we can do to stop them. And we are looking at the microbial angle there. It makes sense that bumblebees have been declining because we have been radically transforming their habitat in a very brief time frame relative to bumblebee evolution. And this is kind of a cool simulated satellite image we're looking at on the left here. Someone made an image of what California would have looked like in a satellite view in 1851 and compared that to a real satellite image in 2013. So you can see how starkly different these landscapes are. You can see, on the right there, the gray blobs around the San Francisco Bay Area and the LA Basin. That used to be green. You can see how much water is in the Central Valley. That is now primarily agriculture. And it's kind of hard to imagine what this was like to be a bee back in this time. But I like this quote from John Muir, who said that the Central Valley was one smooth, continuous bed of honey bloom. And he said, when I first saw the Central Garden, the most extensive and regular of all the bee pastures of the state, it seemed all one sheet of plant gold, hazy and vanishing in the distance. It's kind of hard to imagine if you live in the Central Valley or if you're driving through on the 5 or something, kind of hard to picture that, but it must have been a really excellent place to be a bumblebee. The landscape is very different now. There's still lots of bumblebees. There's still a lot of diversity. It's just a more challenging environment. So my lab is working on how microbes play into this. How might microbes help bees as they're trying to navigate this changing world? And conversely, how might microbes, pathogens and parasites be contributing to bee declines or accelerating the effects of other stressors? A little bit of background on the microbiome of bumblebees. So they have a very simple microbiome. They only have about 5 species of bacteria, way simpler than our own gut microbiome, which has hundreds and hundreds of species. It's very simple and it's very consistent. Hopefully you can all hear me, but okay, sorry. Let me know if the audio gets disrupted, but okay. So they have these 5 species of bacteria, and these are very specialized. They only grow in bumblebees. The names are listed here. You may be familiar with lactobacillus. It's in yogurt and it's on humans. Bumblebees have their own bumblebee-specific cluster or lineage of lactobacillus. So this is what I'm referring to, these 5 bacteria. This is what I'm referring to as the core microbiome of bumblebees. There's some experimental evidence that these core bacteria are beneficial for bees, primarily in providing resistance to disease. So we know that the microbiome provides resistance to Cryphidia bombi, which is a single-cell eukaryote. It's a trypanosomatid parasite related to the cause of sleeping sickness. It's a common pathogen in bumblebees. Bees that have their core microbes are more resistant to it. That's the case for bacterial pathogens as well. And there's some evidence from honeybees that the gut microbiome may also provide resistance to viral infection. And conversely, if you disrupt the microbiome, that may make bumblebees more vulnerable to diseases. And we are seeing very frequently that something is disrupting the gut microbiome of bumblebees. In California, in other places as well, but we're focusing on local species of bumblebees for this research. And I'll give you a little cartoon view of what this disruption looks like, and then I'll show you some data. So normally the bumblebees have these 5 specialized bacteria. I'm calling the core microbiome. It's very consistent, but we also collect bees in the field foraging from flowers. They look the same. They seem healthy from the outside, but when you sequence their gut microbiome, it's completely different. They have very depleted levels of those core bacteria, and they have gained kind of a grab bag of bacteria that are common in the environment, but which aren't specialized to grow in bumblebees. And it's kind of a varied assemblage of bacteria that invade. And I'm referring to this altered microbiome state as dysbiosis. All right, I'm going to show you a little bit of data on what this looks like in terms of, kind of in the bees themselves. So this was a project that I started last summer, toward the end of last summer, collecting the yellow-faced bumblebee, Bombus vasasensii. This is the most common bumblebee in California. I collected a bunch of bees, ran the gut samples through our DNA sequencing pipeline. And on the right, I'm showing the different sites that I collected from, some along the coast and some at higher elevations, and I'll come back to that in a few slides. Okay, so now we're looking at the DNA sequencing results, and here we're looking at what's called an ordination. So this is essentially a 2-dimensional representation of microbiome similarity. Each of these colored dots is an individual bee, and the closer 2 dots are together, the more similar their microbiome profiles are. So you can see that all of these yellow-faced bumblebees, they tend to sort out into these 2 pretty discrete clusters. The blue one on the left, dominated by core bacteria, and the dysbiotic one on the right. And then there's a couple oddballs that don't really fall into either category, but predominantly they form these very discrete clusters. If we look at which bacteria are in which microbiome clusters, so we're looking at the bottom. So you can see that the core bacteria, which are the 5 bumblebee specialized bacterial lineages that I mentioned, they are very abundant in the core dominated bumblebees. They're depleted in the dysbiotic bumblebees. Conversely, the dysbiotic bumblebees are enriched in Pseudomonas, which is a common opportunistic bacterial pathogen of animals, plants. It's really common in the environment, soil, water, pretty much everywhere. And Pseudomonas is almost absent from the core dominated bees. And as I mentioned, there's kind of this grab bag of bacteria. These are other non-core bacteria from the environment that are showing up in these dysbiotic bumblebees, but which are almost absent from the core microbiome bees. We don't know how this is actually affecting the bees, but I have some correlational evidence that it's associated with parasitism. So here we're looking at whether bees were infected with Cryphidia, which is this single-cell eukaryotic parasite of bumblebees, or not infected with Cryphidia. So if you look at the left, bees with the normal core gut bacteria, about 25% of them are infected with Cryphidia, whereas the bees with the dysbiotic microbiome, around 3/4 of them were infected with Cryphidia. And there's reason to believe that this is because the lack of core bacteria is making the bees more vulnerable to Cryphidia infection, rather than the Crithidius coming in and kicking out the core bacteria, but we need to do those experiments to really make sure of that. So I'm really interested in what is driving this. What are the causes? And primarily, is this a natural phenomenon, or is this a result of anthropogenic or human-caused stress? So among the kind of natural explanations, maybe this is just aging. So we know that in humans and mice and fish and flies and lots of animals, the microbiome kind of breaks down in old age, and that makes sense. Kind of a lot of aspects of the body are slowly start to deteriorate in old age, and that includes the microbiome. So maybe these dysbiotic bees are just old bees. When we go out into the field and collect bees, the old bees don't come with glasses and canes and gray hair. It's often pretty hard to tell how old a bee is when you collect it. And so we don't really know. We just sequence their microbiomes and see that they're different. So to address this question, I did an experiment where I raised and measured microbiomes and bees of different ages. And to cut a long story short, this doesn't seem to be the explanation, because even in very old age, their gut microbiomes are super stable. And it's kind of an interesting question of why that is, but that's kind of a story for another day. But both those core bacteria and as well as the strength of their gut immune system are very stable even in old age. So it doesn't seem to be individual aging that's driving this microbiome dysbiosis phenomenon. So maybe it is a result of anthropogenic stressors. And if so, kind of one primary candidate is heat stress. So as I mentioned, bumblebees, they tend to be large, furry, susceptible to heat. We know that bumblebee populations are declining or shifting their range, contracting their range in response to warming. We also know that in many insects, the gut microbiome is particularly sensitive to heat stress. So maybe the heat stress is zapping these core bacteria, and that's allowing the bacteria that we see in dysbiosis, like Pseudomonas, to kind of circumvent that and to infect the bees. So I did an experiment, and to cut another long story short, this doesn't seem to be the explanation either. What I found is that the core gut bacteria are actually quite robust to heat, and that's an interesting question of why that is. They seem to be even more heat tolerant than the bees are themselves. So this doesn't seem to be a kind of an obvious candidate driver for dysbiosis either. But if we look at the spatial distribution of where we see dysbiosis, I mentioned I collected from both high elevation sites in the mountains as well as along the coast. We're seeing something interesting. So if you look at the graph on the top left, what you can see is that around 75% of the bees collected at high elevations had the core microbiome, whereas along the coast, they were all dysbiotic. And this, I should mention, these bees were all sampled around between August and September in 2021. And I'm showing you some photos of kind of the landscape of these different sites, and shout out to the Coal Oil Point Reserve, which is near the UCSB campus, really beautiful spot where I collected some of the coastal bees. So I think, so there's this spatial pattern. I think that's actually a temporal pattern in disguise. So I'll try to explain what I mean by that. Well, we know that phenology, or kind of the timing of the life cycle of organisms, it's not synchronous across colonies of bumblebees across their range. It's very strongly controlled by elevation. If you imagine your bee in the mountains, it's in the spring, you're not going to be able to start your colonies until the snow has melted, until there's actually flowers blooming. So they might get a later start than bees at lower elevations. And conversely, along the coast, the kind of season might wrap up sooner. At least in 2021, we're in the middle of an extreme drought. There are very few bee flowers available at the sites that I was collecting from. So potentially, this collecting window late in the summer was kind of in the middle of the colony cycle for the high elevation bees and toward the end of the colony cycle for these coastal bees. So potentially there's actually a colony aging effect. So this is my new hypothesis. It is natural, and it is a consequence of aging. I mentioned it's not a consequence of individual aging, but it could be a consequence of colony aging. So I'll give you a little bit of info about the bumblebee colony cycle to try and explain a little bit more about what I mean here. So the bumblebee colony starts with a single individual, a single queen called the foundress queen. She does all the work preparing the nest, collecting food, lays eggs, Those eggs eventually turn into workers, and there are successive generations of workers in the colony that do all the non-reproductive labor, collecting food, defending the nest, incubating, brood, etc. At the end of the colony life cycle, reproductives are produced. So there's males, there are new queens that are produced. The new queens mate with the males, they overwinter, and then start the cycle again. So potentially, there's something about being a worker born in an old colony that may make them more vulnerable. So potentially, they're in the same nest, they don't clean that much. Could be that microbes that are coming in from the environment, from flowers in particular, are kind of building up, it's getting dirtier and dirtier. And at a certain point later in the colony cycle, the bees never get their core microbes and instead get these miscellaneous environmental bacteria. So this is a hypothesis and we need to test this. And one key place for doing this is at Valentine Camp. And so this is a photo I took last summer when I was there kind of scouting the area and collecting some bees and evaluating possibility for projects there. So it's a really beautiful spot. I'm really excited to do more work here. And the nice feature of being up in the mountains is the season is fairly short and very discrete. So it's easy to sample from a single population or from a community of bumblebees, because there's multiple species up here, over time to see whether it is actually a colony aging effect. So kind of bringing this back to bee health, bee conservation, how might this information actually be useful? So we think about this dysbiosis phenomenon in particular, there's a couple of different outcomes that we're evaluating. One of which is this colony aging hypothesis. Dysbiosis is a natural phenomenon. Maybe it doesn't strongly reduce bee colony reproduction. That makes sense because otherwise it would have been weeded out by natural selection. This would be good news. Maybe we can not worry about it so much. Maybe we can kind of focus our attention on more pressing microbial aspects of bee conservation. There's another hypothesis, the unknown stressor hypothesis. I mentioned heat stress doesn't seem to be kind of a leading candidate here, but there's a lot of ways that we are affecting the environment that could be impacting bees and their microbes. So maybe dysbiosis is caused by human activity. We don't know what that is yet, so we need to figure out what is the cause and is there something we can do about it? So this would be kind of a really major next step, depending on what kind of data we get. So stay tuned for results. Hopefully we'll have some to share in about a year or so. Feel free to follow me on Twitter if you're interested in learning more. And I wanted to end by talking a little bit about how you can get involved. If you're an undergraduate student, there may be research opportunities on your campus. There may be a professor who's working on bees or a professor that you could convince to work on bees because bees are so cool. And if you're not a student, or for everyone, you can always learn about local bee diversity, tell your friends. It's not quite as popular as birding yet, but I think beeing could be, right up there soon. I really like this book on the left here, The Bees in Your Backyard. It's a really great kind of overview of bee diversity and helps you identify bees. And I also wanted to give a shout out to Am I Even a Bee, which is a fantastic children's book. So if you have kids or if you know someone who has kids, get them a copy of this book. It's really great book about bee diversity and human diversity and identity and so forth. So, and the illustrations are awesome. I also wanted to plug the California Bumblebee Atlas. So this is something that anyone can contribute to. It's a community science effort to track and monitor the state of bumblebee species in California. It's really fun. You get to go out in the field, learn how to identify bumblebees, you collect data, you enter that into the system, and it leads to really important data that's used for bumblebee conservation in the state. So I highly recommend checking out this website. You can get involved before the next season starts in the spring. There's also a lot of stuff you can do kind of on the ground. Supporting policies that help conserve native bee biodiversity is a really big one. The Xerxes Society is doing a lot of great work. This is kind of the organization that's doing the most for invertebrate conservation, particularly bees. You can become a member, you can check out the website. If you have a farm or a garden or a patio, you can plant some bee-friendly wildflowers and shrubs. There's a lot of resources for how to go about this. There's the book California Bees and Blooms, which I like. The Xerces Society also has kind of regionally specific recommendations for what to plant, and they also have guidance for how to make sure you're buying plants that are bee safe, that aren't covered in insecticides. Again, if you have a farm or garden, limiting your use of pesticides and herbicides is really important, as is leaving nesting habitat intact. So we often think about, the food that they need, the flowers, but we don't think that they also have to make nests. They have to have a place to rear their offspring. So logs, mulch piles, dead branches, all of these things can be really important as nesting habitat for a broad diversity of bees, not just bumblebees. And I'd like to thank the members of my lab and collaborators on some of the projects that I mentioned. Special thanks to the UCSB NRS staff for helping coordinate and make the fieldwork possible. Thanks to Cheryl Taylor, botanist, who really helped a lot with bee monitoring and plant monitoring this summer at Valentine Meadow, and these funding sources. And I also would like to thank the many bees that I've had the fortune of working with. And thanks for listening.
Wonderful. Thank you, Toby. That was a very exciting talk. I have a bunch of questions for you, and there's already some questions in the Q&A as well. And I'm just going to give a quick reminder to everybody. Go ahead and type your questions there into the Q&A. Your questions can either be for Carol, our director at the Valentine Camp Reserve, or for Toby on his really interesting talk here too. So I'm going to just go ahead and dig right in. Our first question, Toby, this one's for you. This is from Scott Cooper. And Scott says, fantastic talk, and asks, in your opinion, are documented bumblebee declines owing to the direct lethality of toxins rather than mediated through the microbiome?
Thanks, Scott. That's a really great question. Very complicated question to answer, but I'll do my best. So it's a very important question of, are these stressors affecting bees directly or via their microbiome? In the case of toxins or insecticides, it seems pretty likely that a lot of those effects are direct. The most common chemical stressor for bumblebees, neonicotinoid pesticides, these affect the nervous system of insects. There are experiments showing that they don't seem to disrupt the gut microbiome. So those problems are probably not mediated by the microbiome, but I would also say that there's some interesting work on the herbicide glyphosate. This is the active ingredient in Roundup. It's incredibly commonly used in agriculture globally. And it's been thought that, well, it's not a problem for animals because animals don't have the target enzyme that glyphosate targets. It's only in plants and bacteria. But if you are an animal that relies on bacteria for your health, such as many bees, then you are kind of indirectly vulnerable. So we do think that that glyphosate potentially, and maybe other chemicals too, are indirectly affecting bees via their microbiome. But there's a lot of other stressors, like lack of food, that may also kind of synergize with the microbiome, supplementing or not supplementing nutrients and so forth.
Sounds like there's a lot to work on.
Yes.
So here's another question from Dennis Beebe, and I have the same question as well. And Dennis asks, what is the lifespan of the bees studied, and what is meant by old age? What's an old bee?
Yeah, that's a great question. And I would say that it's very dependent on whether you're studying them in the lab or in the wild. And if you study them in the lab, they can live for up to 3 or 4 months. Pretty long for an insect. Mm-hmm. And interestingly, there's a large discrepancy between the queens and the workers. They're both female insects. They're genetically identical, but they have these very different forms and lifestyles, and the queens live a lot longer. So they can live a few months in the lab, but in the field, they probably almost never live that long because they're gonna, get eaten or die out in the field or something. And the colony lifespan, which I talked about, generally it's a few months to several months, but that really depends on the bumblebee species. So for example, There's a bumblebee, Bombus polaris, which is the only bumblebee that nests north of the Arctic Circle. So it's a very brief window in which it can kind of make a living. And that has, I believe it's like a month, and it only has one generation of workers. So there's a lot of literature on this, and there's some interesting kind of variation among castes and among different species.
Thank you. Okay, so the next question is from Angel Ramirez. And this is, I love Angel's question in part because this is maybe a little shout out to one of UCSB's other reserves, the Santa Cruz Island Reserve. And next week, we actually also have another bee-related project being presented for Santa Cruz Island Reserve. But Angel asks, hi, Toby, have you thought about sampling mating bees, native bees, on the Channel Islands? If you have, what do you predict to see in their microbiome?
That is a great, I have not thought about that, but I love the idea. I haven't actually been out to the Channel Islands, so I feel like I definitely need to go. I'm not even sure what I would predict. It'd be interesting to know how much dispersal there is between mainland California and the Channel Islands. You know, how much of a barrier is that gap? Because if it is a barrier, then you could expect some, you know, because they're isolated, there'd be some interesting kind of evolutionary dynamics on these island bees. And there's a lot of interesting kind of science out there about how organisms adapt to islands in terms of changing their body size and losing or gaining traits. And we don't really know much about how microbes might kind of play into that. Mm-hmm. So I'm gonna have to think about that some more, and I really like the idea, so thank you.
Great. Well, you'll have to come join us at Santa Cruz Island out there. I know somebody who can help you get out there. So.
Oh, great. Yeah, that'd be awesome.
And Angel has a little bit of an add-on. He says even dispersal between islands too, something fun to think about. Mm-hmm. There you go. Okay, so here's another very interesting question. This is from Paul Amaru. Paul, please accept my apologies if I'm pronouncing that wrong. Paul says, currently I am collaborating on a bumblebee project on the Patagonian station of the Pontifical Catholic University of Chile. We are comparing the ecological and biological factors of the Chilean native bumblebee and the introduced one. So the native one is B. dalbomii and the introduced one is B. terrestris. I'm just slaughtering all the names tonight. The introduced bumblebee is a very efficient competitor displacing the native bumblebee. And Paul's questions are, the core microbiome of the two species should be different, or should they be different? The microbiome of the introduced bumblebee could be transferred to the native and affect its life parameters.
Really cool. For those of you who are not familiar with Bombus dalbomii, it's a huge, really beautiful bee, and it's highly endangered. So that's great that you're working on it. Almost certainly, before terrestris was brought to Chile, terrestris and dalbomii had different strains of the same core bacterial species. So probably both of them have Gillymella, Lactobacillus, Nigrosella, et cetera, the ones that I talked about, but different strains, kind of the next taxonomic level down from species. It is very likely that there is transmission from terrestris microbes into dalbomii. We know that bumblebees like to defecate on flowers, and there's a lot of opportunities for transmission of bacteria as well as viruses when one bee visits a flower and then the next bee visits the same flower. So there's a very high chance of that occurring. Whether that's bad, hard to say. You could imagine maybe it's even good. Maybe the terrestrials are coming in with particularly hardy strains of gut bacteria. That would be an interesting experiment to do. So thanks for your question.
Great. Okay. So this next question is for Carol. Carol, this is from Scott Cooper, and Scott asks, what is the status of the bear population at Valentine? So bears, not bees. Does the bear does the bear population remain constant, or does it vary through time?
Okay, well, thanks, Scott. As you know, I'm not a bear biologist, and we don't track the bears at Valentine. We do see the same individuals relatively frequently at Valentine, the same bears, and most of those bears kind of call Valentine their home, and we've seen the same individuals over time. So generally speaking, my guess is that there are somewhere around maybe 5 to 10 bears that sort of call Valentine their home. And I'm hoping that we can bring in one of our local bear biologists actually this spring for a seminar. So stay tuned for that.
Great. Okay. And so the next question here is from Graham Montgomery. And so Graham is with the CEC class. So Graham says CEC class here. So shout out to you guys out there at the Page Center. We're glad you're tuning in. And I just want to take a quick minute to tell everybody what the CEC class is a little bit more. Carol mentioned that before too, but this is a system-wide UC Natural Reserve System class. Stands for California Ecology and Conservation. And there's about 3 students from each UC campus, and all together with some amazing instructors and TAs, they travel across the state and stay at different reserves, and they really learn and practice the process of science from beginning to end. So they're our favorite students out there. And so again, shout out to CEC class. And so after that, Graham asks, Graham has 2 questions, and this is for you, Toby. One, what's your favorite bumblebee? And then the second question is, do you think bumblebee diet shifts due to adventive flower species could this play a role in dysbiosis?
Well, that's first question, very difficult. Probably my favorite is the Morrison's bumblebee, Bombus morrisonii. It's a really beautiful bee, has very short hair. They're black and yellow. They're kind of a desert bee, and you do, at least historically, find them in parts of California, and they're pretty big, and they have short little faces. They kind of short round faces. They're really cool. So Bombus morrisonii, morrisoni. And the second question is about whether kind of changing flowers might influence dysbiosis. Potentially, we really don't know. We're going to be working on that. Actually, we have an undergrad in the lab, Perla Vasquez, who's doing experiments studying how diet and the microbiome interact and affect bumblebee health and nutrition. And so that might shed some light as to whether different flowers of different, nutritional qualities might be causing microbiome shifts and affecting bee health.
So I have a question. I wanted to get back to, you mentioned that bumblebees are furry, and that that furriness maybe makes it hard for them to deal with heat. And can you talk a little bit more about bumblebee fur? What is it made out of? And wouldn't the fur maybe make it easier for them to deal with heat? They could maybe, regulate somehow in a little different way. Maybe you can just tell us a little bit more about this furry aspect of bees.
Sure. Well, maybe getting a little bit out of my element here, but it's sometimes called pile or hair, but it's covering most of their body. There's a lot of bees that are, bees range in how hairy they are. We do see that bees like Bombus polaris I mentioned in the Arctic Circle, it's like a fluff ball. And Bombus morrisonii, which I also mentioned, which primarily lives in kind of arid regions, very short hair. So we do see that hair length tends to vary with the climate of the region that bumblebees are in. And I would also just add that having to take care of brood and thermoregulate their nest is another kind of challenge for bumblebees coping with high temperatures. The brood are very sensitive to shifts in temperature, and bumblebees can fan the nest if it's getting too warm, but they're only capable of cooling it down so much. So it's not just at the individual level that they can be susceptible to heat, but also at the colony level, thinking about offspring reproduction.
Great. Okay, so there's a couple more questions. First, there's a comment that I just wanted to pass along from Barry McPherson. Barry says, great talk. Good luck with your research. And Barry used to be a Snarl seasonal employee from the late 1960s and currently lives in Mono County. So Barry, thanks for tuning in and look at what your work has done to produce up here at Snarl and the Valentine Reserves. Okay, this next question is from Bobby Offen, and Bobby asks, in general, why are we always worried about being stung by a bee? Is this a real concern with the bumblebees?
It's a good question. They're not aggressive. You don't really have to worry about it unless you are like me and you're, rearing them in the lab and you're messing with them, or if you accidentally, like step on one or something like that. In general, it's really not a problem, but they will sting you if they are threatened. So if you grab one on a flower. However, I should mention that the males do not sting. So the stinger is a modified ovipositor, egg layer. And so this is essentially an apparatus that only female insects have. So the males can't sting. They kind of pretend like they're stinging you. They kind of move their abdomen and it can startle you, but they can't sting at all. So it's, if you learn how to identify males and females and you're confident about that, you can just grab a male bee off of a flower and it's kind of a fun party trick. But I would make sure that you're very confident in your bumblebee identification before you try to do that, because otherwise you will be stung. It's not so bad though, the sting. I don't think it's as bad as a honeybee sting. I tend to swell up, but it's not like incredibly painful.
Interesting. Okay, so I think we have time maybe for one more question. And this is an interesting one too. Again, from Dennis Beebe asks, aerodynamicists years ago might have predicted that bumblebees could not fly. And he likens this to the old C-124 cargo aircraft. Are bumblebees worse flyers than other bees?
Yeah, this is a great question. And also great name, Dennis. It's true. So a long time ago, it was thought that based on the physical properties of insect wings, bee wings, they shouldn't be able to fly. But it didn't account for flexion. And I'm not an engineer, so I may be getting some of the terminology wrong, but it didn't account for the flexibility of bee wings. It's giving them a lot more lift or something. And there is a lot of interesting work on bumblebee flight biomechanics. I would encourage you to check out Stacy Combs's work. She's at UC Davis. She's doing a lot of really cool work on bumblebee flight. She has a wind tunnel in her lab and lots of really cool work going on there. I don't know about the kind of comparative flight performance of bumblebees relative to other insects. They are larger. One kind of advantage is that they can fly in much colder conditions than honeybees or other bees. This is where it's advantageous to be large and hairy. They can also, they're not strict ectotherms like, most reptiles and insects and fish and so forth. They can generate their own body heat. So that allows them to kind of warm up and then get to kind of flight temperature before they go out into the environment to forage. So you can see bumblebees foraging at much lower temperatures than you would see any other bee foraging. So that is a kind of a good trick that they have.
Excellent. Well, I think that's all the time we have for this evening. So I want to just first tell you all about the exciting talk that we have next week. We will be hearing from Dylan Travis, who's from UC San Diego, a PhD candidate in the biological sciences division there. More bees, everybody. So the title of his talk is Gauging the Impact of Abundant Non-Native Honeybees on Native Pollinator Communities, a mainland island comparison. So on health, if you're out there, tune in next week. This will come to you from the Santa Cruz Island Reserve. And with that, I just wanted to give a big thanks to Carol for introducing us to Valentine Camp Reserve, and to Tobin Hammer from UC Irvine for such a great talk tonight. And for all of you out there, thanks for your great questions and for tuning in. And we look forward to seeing you next Tuesday. So thanks, everyone. Have a great rest of your evening.
Thanks, everyone.
Thanks.